Hello, everyone. My name is Mel Afrime, Executive Vice President, Translational Medicine at Altasciences. I'm delighted that you have given me the opportunity to share Altasciences' expertise and insights in support of multi-regional drug development, or what was also termed global drug development, back in the 90s. The title of my presentation is Ethnic Bridging in Phase 1 Clinical Trials, a strategy for enhancing asset value and accelerating global drug development. I'll share with you several topics today. I'll start with discussing the drug approval lag in Japan during the 90s, how that led to the ICH E5 guidance. Then I'll discuss several examples of what we call the ethnic bridging paradigm and how that led to ICH E5, E17 guidance, which discusses multi-regional clinical trials and how to evaluate and launch those trials, and then share with you some bridging trial acceleration strategies that we've employed over this past several years, and then provide a summary, and then move on to questions and answers. Let's discuss the drug approval lag in Japan. During the 1990s, it was apparent in eastern regions of the world that drugs were not getting to the market as rapidly as those drugs in the western regions of the world. For example, Claritin, a drug that I worked on the development while I was with Schering-Plough, was approved in every Western country by 1993. However, it wasn't approved in Japan until 2002. And that approval was hastened by taking advantage of the ICH E5 guidance. Claritin was, you may be aware, approved for seasonal allergic rhinitis and chronic idiopathic urticaria. Another example is Doxil, a drug for ovarian cancer and AIDS-related Kaposi sarcoma and multiple myeloma, was available in the West in 75 countries by 1999. However, wasn't approved in Japan until 2009, and that also was approved based on the ICH E5 bridging strategy. Similar examples could be cited for both new indications as well as post approvals, and for both drugs developed in Japan and those developed in other countries. This drug lag was viewed as a major public health issue, and overall it was noted that in Japan it took about 3.8 years in average for the review period for a drug to be developed, whereas in the US and for the Western countries, it was just over a year for the approval process and launch of new chemical entities. I'd like to review some of the possible reasons cited for this drug lag in Japan primarily. One reason cited was sluggish drug innovation. The prescription drug health insurance reimbursement required a lengthy review, and prices were lowered biannually. And that was determined upon approval of the initial price. This created pressure to develop low-risk drugs in order to realize the profits with the least upfront investment, which also created an immature entrepreneurial environment in that country. Overall, clinical trials were seen to be quite expensive in Japan, almost 4 times the cost as in the EU or the US, and there were also a lack of seasoned principal investigators in Japan at the time. The drug approval process also had delays. Navigating the system was difficult. There were language and cultural issues. Even sponsors who were developing drugs in Japan had to go through an accreditation process as a foreign manufacturer, even to bring their drug into the country. And the whole process was evaluated under the pharmaceutical affairs law, not primarily a medical or scientific review. And all forms had to be in Japanese. Also, overall, it was thought that the PMDA was understaffed. So they rapidly, at that time, developed a plan. They wanted to modify the drug pricing system. Certain drugs under patents were exempted from the biennial price reduction. The MHLW identified 109 prescription drugs to receive prompt reimbursement decisions. 91 new chemical entities were identified that would receive expedited development by both discussions with pharma and through the MOHW. Then 200 Japanese institutions were identified that could prescribe unlisted drugs in order to meet identified unmet medical needs. And furthermore, the ICH supported the ICH E5 guidance, accelerated bridging of drugs which could have had full approval in other regions, and the Japanese participation in global drug development programs or multi-regional clinical trials. Let's look at the impact of these early decisions and to help shorten the review and approval process in Japan. Looking at the review time in Japan relative to the review time in the U.S. over 2006 to 2009. Looking at the pre-application timing and the review timing, the totals were about, let's say, 2.4. There was a not so good difference in 2007, but 2008 and 2009 showed a downward trend to the timing and some suggestion that there was a positive impact on these decisions, how to move things forward and get drugs to the population in the eastern region a bit more rapidly. In September of 2007, the PMDA published an official document where they demonstrated official acceptance of the ICH E5 bridging strategy and supported utilizing the bridging strategy for global clinical trials in an effort to avoid having this unacceptable drug lag continue in the eastern region of the world, primarily in Japan. Just to review what was seen to be occurring worldwide and what was part of the basis for the drug lag in Japan, we see that development was carried on in silos worldwide. In the US, an IND was submitted, the first in human trials started, and then phase 2 and phase 3 occurred primarily in the US, sometimes in the US and the EU or South America, but it was developed in silos. In Japan, the IND was submitted, it could be at a similar time. First in human trials, including the SAD and the MAD studies, and phase 2 development, phase 3 development occurred within their own time frame based on data that was integrated from these studies, and the process moved forward to the review in that country. It was a similar picture in other regions of the world, in South America and alike, and it was seen to be the main root cause for the different timings in approval throughout the world, primarily in the eastern region of the world. At this point, I'd like to review some of the basic tenets of the ICH E5 document. The guidance provided a strategy to bridge data and information from a fully developed dossier in one region of the world to another region of the world which hadn't started development or had premature development of that same chemical entity and hasten the approval process. It was meant to minimize duplication of clinical data. It provided bridging requirements for extrapolation of the foreign clinical data to a new region. And it also suggested that global drug development or multi-regional drug development was a clear strategy to avoid reliving the profound drug lag or difference in timing for launching new medicines throughout the world. The extrapolation or bridging requirements that were cited in ICH E5 required a complete clinical data package. It required adequate characterization of the pharmacokinetics, the efficacy and safety. A clear definition of dose response was discussed, and they also had to review the acceptability of the evaluation of the clinical disorders for that region. And then after these concepts were evaluated, they would determine the requirement for bridging strategy. It was a very concise and clear presentation of how to bridge the development and results of development from one region to another region of the world. It was concise in that if you followed the 3 steps, that is, assess the completeness of the foreign data, assess the product sensitivity to ethnic factors, and then judge the requirement for a bridging strategy, you would have a clear answer of whether a drug could be bridged from one region to the other or not. These ethnic considerations, for the most part, relied upon the drug exposure or the pharmacokinetics, and then how this would lead to evaluation of safety, tolerance, and efficacy or dose response in one region, and the likelihood of having this same behavior or pharmacokinetic or exposure of a drug be similar from one region to the next, and predict whether it would likely be similar in one region of the world versus another. The pharmacokinetic determinants were seen in 2 lights. One, extrinsic factors or cultural or behavioral determinants of a drug exposure, or intrinsic factors, that is, the genetically driven factors that would impact the exposure or the ability to extrapolate from one region to the next. Let's review these ethnic considerations in a bit more detail. As I said, intrinsic factors are genetic or, let's say, physiologic or pathologically determined in an individual, whereas the extrinsic factors are somewhat environmentally determined. And then looking at those categories, you could see that height, weight, hepatic, renal function, race maybe could be determinant. Stress, some things could be considered to have impact on both intrinsic and extrinsic. Stress could turn on some genes that may or may not be active under various non-stress conditions. Receptor sensitivity is genetically determined, whereas medical practice in one region is considered extrinsic. Some people may practice medicine according to various culture pressures versus what we would consider scientifically defined parameters for medical practice. Other things straddle extrinsic and intrinsic, smoking, alcohol, dietary habits, perhaps, can straddle the two extrinsic and intrinsic determinant factors. Specifically, from a pharmacokinetic perspective, we want to know if the drug is linear or nonlinear. We want to understand the concentration effect curve. We want to understand the pharmacokinetic, pharmacodynamic outcomes, that is the therapeutic margin. Metabolism, is it polymorphic? Is it extensive? Is it a prodrug requiring rapid conversion to the active moiety? What is the extent of plasma protein binding? Is the drug used with other medications? Are there multiple anti-infective agents, antiviral agents? In psychotherapy, are there multiple drugs required for therapy, and how does each one impact the outcome of another, specifically from one region to the next? And what is the likelihood of inappropriate use or abuse of the compound in one region or the next? I'll spend the next few moments reviewing with you how regulators actually used ICHE5 to help curtail the drug lag back in the 90s. Let's look at a high-level decision tree on utilizing the ICHE5 concept. If a medicine were ethnically insensitive, that is, that there was no change in the exposure of a drug depending on the region of the world, and medical practice throughout the world were similar, a drug could be bridged. That is, no particular bridging studies would be required based on the ICHE5 concept. For an ethnically sensitive drug, if the ethnicity of the region you were wanting to bridge to were similar and clinical experience was adequate with that medication, again, the drug was open for a determination of no bridging studies required to approve that drug in another region. For an ethnically sensitive drug, that is, there was some concept that exposure could be different in one region to the other, and the ethnicity of the region was different. That is, let's say, the classic would be from a Caucasian mainly to an Asian population, but medical practice was similar, and there was familiarity with that drug class. The drug could be approved solely based on a pharmacokinetic, pharmacodynamic characterization of the drug in the new region, enabling approval based on characterization. The most complicated case would be that there were doubts about the choice of the dose, that is, the dose ranging, dose response data seemed to be different in one region to the next, perhaps medical practice being different. It was a relatively new drug class. Clinical experience in that region was insufficient. The evidence would point to the need for a controlled clinical trial or full development in order to get the drug approved in the new region. Just based on this, you could see that use the guidance appropriately, there would be prevention of unnecessary duplication of clinical trials. You would have a more efficient and cost-effective drug development process in a global basis. The drug lag issue would be very much less apparent, and it also would support global, simultaneous, or multi-regional drug development. The ICH recognized that the ICHE5 in the suggestion that global clinical development or multi-regional drug development was a fundamental solution to the drug lag problem, and therefore published ICH E17, which was further guidance on the principles for planning and design of multi-regional clinical trials worldwide. The ICH guidance depended largely on the ICH E5 factors discussing evaluation of foreign clinical data, but it also stressed the need for good clinical practice, general considerations for clinical trials, statistical principles in how to define sample sizes in a global clinical trial, control groups in global development of a new drug or new drug class, and the pharmacogenomic management of data from such trials. Multi-regional clinical trials conducted according to the guidance were seen to allow the investigation of treatment effects, that is, safety evaluation, potential impacts of the intrinsic and extrinsic factors on exposure, and that it was seen to facilitate efficient drug development clearly across the world. In essence, there was a potential to enhance scientific knowledge about how treatment effects vary across regions and populations under the umbrella of a single study protocol, and how this variation may be explained by extrinsic and intrinsic factors of drug exposure. In this slide, I review the results of a 2020 publication in Clinical Pharmacology and Therapeutics, which reviewed the median drug approval lag from 2008 to 2019. And you see that there definitely was movement from as high as 6 years to progress downwards to somewhere less than 2 years at 2018-2019 time period. Clearly progress and clearly movement towards elimination of the drug approval lag. Another example of that is if we look at the timing and the availability of simultaneously review in the EU, the US, and Japan. We see that it takes 13 to 15 months for the review process in the EU, about 15 months in the US, and the data would suggest that it's even a bit more rapid in Japan. Let's discuss some basic requirements for launching a multi-regional clinical trial or a global clinical trial. What are the basic requirements to conduct a global clinical trial? The first basic requirement is to understand the exposure of the drug in one region compared to the target region. That is, if a drug were developed primarily in the US or phase 1 had been carried out in the US, what does phase 1 look like in the target population? This answer can be defined by a pharmacokinetic trial comparing the exposure of the drug in one region versus the next. What are the basic points to consider in designing your trial? That would be just the same points that you would utilize for designing a phase 2 or phase 3 trial in any region of the world. You just have to understand what the sample size is required for each region where you want to launch that clinical trial. Some considerations for the evaluation of exposure. if the multi-regional clinical trial is going to be launched in many Asian countries, whether it could be China, it could be Korean, it could be Japan as well. You would want to understand what the pharmacokinetics are or the exposure is from some individuals from each one of those countries. And you want to understand, based on that phase 1 trial, if there's some importance in management of concomitant medication based on how medicine is practiced in those other regions. A simple way for Japan is to have a consultation with the PMDA and discuss your plans for launching and what information you have at the point of your consultation. The PMDA will give official advice. That is, if you reach an agreement with the PMDA for what your global clinical trial should look like, they will abide by that decision. It would be considered binding. There is a fee for the consultation with the Japanese health authorities. Just as I mentioned, looking at how ICH E17 guidance has been performing and what information is available at this point for affecting of the individual tenets of the ICH E5 guidance, we see that by and large there was compliance with GCP standards. There was, in many instances, a consultation meeting prior to launching. However, there were 40% of the cases where they were launched without consultation. But one important thing to see that I point out here is that in over 90% of the cases, a comparative pharmacokinetic study was carried out between Japanese and non-Japanese populations prior to launching of the multi-regional clinical trial. In fact, in about the 8 or 9% of studies that didn't require these trials, there was, in some instances, available data already in that Japanese population, or the drug was seen not to require that comparative study, whether it was a monoclonal antibody, a biologic that was considered to be universally bioavailable, and there was really no concerns that dose response would be different in one region of the next. So this is an important fact to understand, that in consideration for launching your global clinical trial, you're going to want to understand the exposure medication in the target region before you go ahead and launch your clinical trial or launch your plannings for that global clinical trial. Let's talk about some acceleration strategies for your development and how that could look like as you move forward. Several sponsors that we deal with just have a vision of developing drugs in the US and Japan, and then considering other regions of the world as a next step in development. For those companies, we suggest doing phase 1 in the United States, following phase 1 or embedded in phase 1, evaluate exposure of the drug in Japan, then perhaps move on to dose ranging studies, dose response studies, and phase 3 controlled clinical trials, but simultaneously use this Japanese, what we call bridging pharmacokinetic data to support launching of phase 2 and phase 3, either in a 2-region phase 2 and phase 3 or simultaneous region, but eliminate the first-in-man and SAD/MAD studies in Japan. Your SAD/MAD design could also be enhanced by including some individuals in each of your SAD or MAD cohorts and even get earlier signal for whether bioequivalence or bioavailability is similar across regions of the world. If you include Japanese or Chinese or Filipino individuals in each cohort, you would get a simultaneous signal whether there is a difference or not in exposure in people from those regions of the world. The paradigm for your decision tree would be if there is a pharmacokinetic or pharmacodynamic signal present, let's say no, there's no signal. You can initiate your multiple dose study. You could include some people from the same ethnic groups and evaluate it under multiple dose conditions, and then plan for your multi-regional or global clinical trial. If you do see a signal, you could include the same ethnic groups as staggered dose group, depending on what the difference was, whether it was enhanced exposure or less than enhanced exposure in that target population. But it's always best to have more information than less and understand what exposure looks like in that target population before you design your global clinical program. And here's what it could look like in a real world, extensive global clinical development plan. Just as I suggested, you do your bridging trial, you do your single dose or multiple dose determination in the US. You understand what your target population is or your target region for development. You could then have consultation with these areas, Japan, Korea, Taiwan, and have a discussion on the strategy for your multi-regional trial. Carry out this development either in as two regions, that is the US being one region for your NDA submission, or carry out a one large global clinical trial based on your consultations with the various health authorities. China could be involved in that decision tree as well. Up until now, China has been accepting this bridging or ethnic bridging data for their decisions to move forward with participation in a global clinical trial. They have been wanting sponsors to repeat phase 1 during the development of the drug in China, but over the past several months, we've been getting signals that they're not asking companies to repeat phase 1 if phase 1 was clearly determined in another region and there's no difference seen in an exposure based on intrinsic and extrinsic factor evaluation. Just some information about the documents required for submission for your multi-regional clinical trial. Applications form are required to be in the language of the country where you're submitting. In those eastern regions, the protocol, Japan, should be in that language. It should also, if it's going to be submitted in Korea or China, it should be in those languages. The investigator brochures should be in the language of the target country. We're used to the US where the CRF sample is required. It's not mandatory in Japan, Korea, and China. And the investigator's CV are not required in Japan, Korea, and China for submission of your INDs in those countries. I just want to review some safety information from, in this case, Everolimus and Temsirolimus that points to a little bit of a validation of a concern, and one of the fundamental reasons for the drug lag back in the 90s. There was concerns that safety was somehow different in the eastern regions of the world versus the western regions. That is, that the Eastern Asian populations was somehow a bit more sensitive to the effects of the drug and would have more adverse experiences at lower doses than in the West. And I use this as an example because looking at accumulated safety data for these medications in Japan, there was 26 or 27% reporting interstitial lung disease in these cancer patients, whereas overseas there was only 12% for Everolimus. For Temsirolimus in Japan, there was a 55% reporting of interstitial lung disease following Temsirolimus. In China, similarly 55% reporting. Korea 64%, but in the U.S. only 30%. Similarly, almost twice for Everolimus and Temsirolimus of reporting of this adverse experience. I do point out that the low numbers, 11 of 20, but it is a valid signal and some cause for the feeling that perhaps there are some adverse experiences where any region could be a bit more sensitive to those adverse experiences, which could be demonstrated with any compound. And it could be based on exposure, it could be sensitivity of the drug, it could be some extrinsic factors which lead to the expression of these adverse experiences from one region to the next. And it's something that we should all be aware of. At this point, I'd just like to review some cases which point out some of our experiences in carrying out these, what we call ethnic bridging studies. That is, that fundamental evaluation of exposure in the core or the first studied population versus the target population. And here in this study, we evaluated 45 Han Chinese, Japanese, and Caucasian subjects, a total of 135 patients or participants. We recruited 100% of the protocol required individuals. Screening duration was 35 days. The enrollment duration was 17 days, and we were able to provide the information to the sponsor to submit their INDs or have their pre-IND meetings and determine the doses they wanted to use in their multi-regional clinical trial at the timing that supported efficient progress of their multi-regional. My second case is one where we studied a drug for glaucoma. So it was an topical ophthalmic administration. There were several ocular assessments, that is, local tolerance of the drug as well as intraocular pressure. 30 Japanese participated in the study. We achieved 100% of our recruitment requirements. Screening was from day minus 21 to day minus 2. They were confined from day minus 1 to day 6. There were outpatient visits on days 4, 7, 8, 16, and 25 or 35. And again, we were able to get the results of the study to the sponsor to launch their global trial in Japan. And actually, it was expanded to Korea and Taiwan. For my third case, I just wanted to give an overall outcome example where we studied several medications for a large global sponsor of ours. 22 bridging trials were carried out on 16 new chemical entities over the past 5 years. 9 of those compounds have received PMDA approval. 2 compounds are currently in phase 2 development in Japan, and 5 await the sponsor's regulatory action. So we started at the top of our funnel here with 22. We ended up with 11 compounds whose data was accepted by the PMDA. Those other compounds are awaiting pre-IND meetings or are waiting a decision whether the sponsor requires development in the Eastern region based on internal decisions. In summary, multi-regional clinical trials are seemingly the norm. Development is progressing as I described, where a single dose or multiple dose ethnic sensitivity study is done to evaluate exposure as a seed study for regional development and participation in a multi-regional clinical trial. The global trials have become quite diversified. There's multi-regional clinical trials among the East Asian countries. There's more early phase and more large-scale trials that are carried out, so that the multi-regional early phase studies are carried out, and the phase 3 trials are on a very large scale. Unfortunately, document translation remains to be an issue, but I'm sure that will be overcome as multi-regional trials continue to be the norm. Thank you so much for your participation in this webinar. There are some questions in the chat box right now, and I'll just move right on to the ones that I have available. Number one, what is the definition of Japanese or Chinese when you conduct ethnobridging? We define the Japanese individuals based on having parents and grandparents of Japanese descent. We always look at individuals' passports to identify that they're the people that they say they are, and then do a family tree and determine that their parents and grandparents from the maternal and paternal side are indeed Japanese or Chinese or whatever the population is that we're studying in that particular protocol. Number two seems to be from the same individual. Further to your first question, can you comment on the qualification of participants with regard to first generation entering the U.S. and how long they can be in the country and still qualify for participation in the trial? Interesting question, and thanks for that. We determine, along with the sponsor, the inclusion criteria and really what the definition is for the participants, whether regards to first generation and how long they are living in the other region, in our case in the United States. And it depends on the sponsor by and large. The Japanese health authorities are less concerned about this. They feel that the most important factors that determine the exposure are the genetic or the intrinsic factors. So as long as, from a Japanese perspective, the Japanese health authority perspective, as long as the participants are first generation, they could be in the United States any time, and they don't care as long as their parents are Japanese, maternal and paternal grandparents as well being Japanese, and they're satisfied. Some sponsors have some qualifications. Some sponsors want first generation who haven't been in the U.S. for over 5 years. Some allow 10 years. but that's a point for consideration when we meet with the sponsor to design the trial. Third question, would you say that if Japan approves a drug that there is a delay in approval from Western countries? Right now, I think that there's no apparent drug lag, that the time for approval is similar in Japan than any other country in the world. Next question. What do the Asian regulators generally request prior to instituting a multi-regional clinical trial? Primarily, the Asian authorities, that is, let's use Japan, Korea, Taiwan, China, like to see that exposure comparison, that PK study that evaluates exposure in the target population. That would be comparing the bioavailability of Japanese versus non-Japanese, Koreans versus Caucasians or non-Koreans, as well as Chinese versus non-Chinese individuals. Foremost, that's the one thing that they require. And then there's individual data that would be driven by the behavior of the drug in and of itself, the target population, the target indication, that they're all would be individually considered. But the most important request is that single dose or multiple dose bioavailability comparison. Next question. Please comment on the need for single dose results versus multiple dose results in sponsor plans for Asian bridging. That's a complicated question. By and large, the most important evaluation in our minds is a single dose comparison. It could be a single dose high dose, a single low dose study. Most important is to have bioavailability comparison at the dose which is targeted for the multi-regional clinical trial or at the therapeutic dose level. Multiple dose studies would be required based on the pharmacokinetic behavior of the drug. If it's a nonlinear drug, you may want to see multiple dose studies. If it's a very variable bioavailability agent, you may want to use perhaps a larger population or multiple dose conditions. But a single dose study generally at least the target therapeutic dose is the primary design for those trials. So that's all the time that we have right now for questions. I'll personally answer all remaining questions after the webinar. If you do have more questions, please feel free to reach out to me at the email listed on the slide. And I would like to say thank you for joining me today and wish everybody a good rest of their day. Thanks so much.